Assalamualaikum and hi everyone. My name is Siti Rohaliza and I am a PhD student from Faculty of Medicine UiTM Sungai Buloh. In this video, I would like to introduce to you our innovation project, the Tech Tracker ELISA Edition. In laboratory research, especially in clinical and biomedical research, ELISA is one of the most widely used techniques to quantify protein expression. And usually, the accuracy and the reliability of the data depends on our technical skill. But we must know that how we keep and document the data is also important. Unfortunately, based on our daily laboratory experience, we notice a few main issues on this. First, researchers record their data on different logbook, loose paper, and also in different Excel files. This will make all information difficult to trace. Second, important details like reagent preparation, plate layout, incubation time, and sample coding are often not recorded in a standard way. All of this will make it very hard to verify the data and write a report properly. So, this is where we introduce our innovation project, the Tech Tracker ELISA Edition. Before we go further, let me explain how this book was developed. First, we assess the need based on our daily laboratory experience. Next, we analyze the existing documentation method, including personal notebook, loose worksheet, and spreadsheet, to understand the limitation. Based on this analysis, we design the structure and content of the Tech Tracker ELISA Edition. After that, we developed the first prototype of the logbook and tested it in real laboratory setting. Finally, feedback from users was collected and improvements were made to produce the final version of this product. Now, let me show you what inside our Tech Tracker ELISA Edition. First, this is the cover of the book, giving you a quick idea of its purpose and design. Inside the book include a guideline section that explains the basic steps of ELISA along with useful tips for each step. Next, there are two main types of pages in this book. The first page contains a 96 well plate where you can record your sample in triplicate. Below the layout, there is a calculation section for reagent like wash buffer or antibody solution. This helps make your experiment more accurate and organized. The second page shows the workflow step by step where you can tick off each step as you complete. Beside the workflow, there is a discussion section where you can write your result and notes. Overall, this book has 100 pages, enough for 100 experimental rounds, and the page and workflow can be edited and customized based on your laboratory needs. Now, let's go to novelty. To our knowledge, this is among the first ELISA-specific tracking logbook designed to standardize documentation across the entire workflow. Unlike conventional lab notebook, this book is not just for writing results, it integrates experimental layout, reagent calculation, workflow tracking, and discussion in a single structured system. By providing dedicated templates for ELISA plate mapping, calculation of buffer and antibody, and step-by-step workflow monitoring, this logbook transforms how ELISA experiments are recorded. From fragmented method into a complete traceable and reproducible documentation system. From academic point of view, first, it will support high-quality research reporting, and then it also can be used as standard learning tool for ELISA documentation, and it also can improve data traceability and reproducibility. Besides the academic contribution, this logbook also have meaningful impact on researchers themselves. First, on academic side, it was easy to use, save time, and we also no longer to scramble over the newspaper. On a larger scale, this innovation can benefit society and the nation. When research data is well documented and verifiable, it leads to higher quality scientific output which can support better healthcare study, inform policy, and overall progress in biomedical research. Building on the success of our previous version, the RNA and the PCR edition, which received a Silver Award for innovation, this ELISA edition continues our commitment to improving laboratory documentation. To conclude, this book is not just a logbook, it is a complete tool to organize experiment, track workflow, and improve accuracy. It helps researchers work smarter, ensure data integrity, and support innovation in both academic and industrial labs. Thank you.